We're picking up with the second video lecture for Chapter 17. This figure from your book is not for memorization. It's only meant to give you an overview of what the CDC's current recommendation for immunization for the population is. Probably it's already outdated, as it should include the COVID-19 vaccine. It's likely that future versions of this schedule will include this vaccine. What you can see over here on the left are all of the different vaccines that are recommended for the general population. The green bars going horizontally across represent when the different doses for the different vaccinations should be administered. It includes the recommended boosters that are needed to occur periodically over someone's lifetime. For example, the DPT, aka DTaP or Tdap vaccine, which includes protection against diphtheria, tetanus, and pertussis, has 3 doses that are to be given in early childhood up to 6 months. You should already have 3 boosters during early infancy, and then another booster will occur a little bit later at 18 months, and again at about 6 years, and again at about 11 years old. Then, as you get into your adult years, you should boost yourself every 10 years to keep the immunity high. This is just an example of one of the vaccines that needs to be given frequently over your lifetime to keep your immune system primed and you protected. You can contrast this with the influenza vaccine, which we know is actually a slightly different vaccine every year, but it's still a flu vaccine. You need to get that annually because it's really against different strains of the virus that will be circulating at any given time. More on the influenza virus and the vaccine in a future chapter. This figure just provides you an overview of what the vaccination schedule looks like. You should notice that there are quite a few immunizations that are occurring in childhood because we're trying to protect against diseases that children may be exposed to and are at greatest risk for serious infections. Immunizations given during early childhood will protect them as they are growing up and can provide good immunity throughout their whole life if the appropriate boosters are given. People are often concerned about vaccine safety, but the things that people are concerned about versus the true risks are often not in good alignment. Overall, vaccines go through quite a rigorous set of clinical trials before they're released to the general public. You may have heard of the phases of clinical trials in the news. Even with Operation Warp Speed, which greatly sped up the COVID-19 vaccine process, the steps were still conducted as usual and as required. The data review and approval process, as well as the simultaneous ramp-up of production of the promising vaccine candidates prior to actual approval, enabled a greatly streamlined, sped-up process compared to the routine approval process. This happened due to the coordinated cooperation between scientists, companies involved in production of the vaccines, and government agencies, and was unlike the typical process. While some people are concerned that the process was rushed and are hesitant to take the vaccine, the testing and safety protocols were not rushed, only streamlined. This only could happen with the willing cooperation of all involved parties and financial support of the federal government that removed the financial risk to the companies involved. Companies normally do not ramp up production of vaccine doses until they have approval, and approval processes normally move slowly. There's not an urgency to this process, normally. Companies cannot shoulder the financial burden of this process if the vaccine is not approved or is not approved after a very long review, such that manufactured doses have already expired prior to approval. In the case of COVID-19 vaccines, the government ensured that the production would be paid for and the companies would not have financial risk. This was the atypical part. The streamlined cooperation between companies and the federal agencies. The concern about vaccine safety is misguided compared to the risk of infection or spreading the infection to vulnerable individuals. So, what are some of the real dangers associated with vaccines? Typically, an adverse event is mild toxicity, meaning you might feel a little under the weather. You might have a little bit of soreness at the site of injection, but this is nothing serious. there is a rare risk of anaphylactic shock, and that's always present in the minority of patients. If you're allergic to some component of the vaccine, the risk of anaphylactic shock is real and something to be concerned about. That's why they try to anticipate what somebody could possibly be allergic to and warn people. For example, when we talked about manufacturing the flu vaccine and growing the virus in high numbers in embryonated chicken eggs, if you're allergic to eggs, then that's a problem. Even though they try to purify the vaccine components, what if some of the egg components got through that process and there's still some egg protein left in the vaccine? Anaphylactic shock is a risk, and they have to be aware of it. Residual virulence from the attenuated viruses, we already talked about that. With the attenuated polio vaccine, there is a risk of reversion or accumulated mutations back to a virulent form if it's passed from person to person often enough. That happens if very few individuals are protected against polio in a population and a few are given the attenuated vaccine. Nowadays, scientists really try to genetic engineer in specific controls into vaccine strains to prevent any type of virulence from reemerging in the attenuated strain. There's been allegations that vaccines cause autism, diabetes, asthma, all sorts of conditions. However, there has been numerous studies from numerous different scientific organizations which have looked very carefully at the data. Even the FDA revisited the data and performed numerous analyses to determine if there was any correlation that could be linked to vaccines. To date, there has been no substantiation to this allegations. It does not appear that vaccinations have any link to any of those or any other particular condition. Remember, correlation does not equal causation. That's one of the major concepts that people have a really hard time with. People want to understand why something occurred. And since vaccinations occur often in childhood, when other conditions are being noticed and diagnosed, it's often correlated with the vaccine. People then allege that the vaccine is causing these conditions, but deeper analysis of larger populations don't reveal that relationship. The federal government set up a vaccine adverse event reporting system. Anyone, including physicians, can log in and report anytime there is an adverse event that occurs within a short period of time after a vaccination. This is monitored closely. If there are a few of these events popping up that seem to be even slightly statistically relevant, they will stop the administration of the vaccine and investigate and pull the vaccine if need be. This occurred briefly during the administration of the Johnson & Johnson COVID-19 vaccine. The vaccine was released, again with a warning, to women to look out for specific symptoms that may indicate an adverse blood clotting reaction. However, this type of vaccine withdrawal has occurred before and illustrates that the system is working. For example, there was a human vaccine on the market for a short time for Lyme disease. It has been pulled because of adverse arthritic symptoms experienced by more than a few individuals after receiving the vaccine. Also, there was a first version of the vaccine against a rotavirus, a diarrheal disease affecting children. An unusual intestinal condition was related to this vaccine and, although rare, was statistically relevant and a serious complication. The vaccine was pulled from the market, revised, and resubmitted for approval. The second version did not have any of those complications and now is widely used to protect children all over the world. The system does work if there's a problem with the vaccine that doesn't show up in the earlier studies. The clinical trials leading to a vaccine's approval involves tens of thousands of people, but once the vaccine is released to the public, millions of doses are administered. When you have that many more people, you might start to detect rare adverse events. So in sum, the Vaccine Adverse Event Reporting System is a government-sponsored system. It's in place to help monitor vaccine safety after the clinical trials demonstrating safety occurred and the vaccine is now being administered to the general population. The system is accessible to anyone with the internet. You don't have to be a physician to use it. If you experience an adverse event near the time you receive a vaccine, you or your physician can report it. If there are a few of these events reported, the FDA will review this carefully and pause or revoke the approval of a vaccine if the data indicates a statistical linkage to the vaccine distribution. Next, we're going to talk about herd immunity. Herd immunity has been discussed a lot in the news in relation to COVID-19. Let's go through it so that you can truly understand the concept because it's sometimes misunderstood. Up at the top panel, you can see that this is more of an unvaccinated, unexposed population scenario. You have all of these blue people, which are susceptible to an infection, and the red individuals are infectious. The red people are potentially spreading that infection to a wide number of people in the population. It spreads quickly in the population. So here you see all those red individuals. Now, very many people are infected. That's basically the pandemic scenario. There's very few immune people and very many susceptible people. If the susceptible people come in contact with infectious individuals, the infection spreads rapidly. Herd immunity is what's shown on this bottom panel. If we have enough people that are immune to the infection, whether that be from normal infectious processes or vaccination, we have a herd immunity situation. Here, the yellow individuals are protected. They are immune. The blue individuals are individuals that are still susceptible. And the red individuals are again infectious. If there's enough yellow individuals, the immune individuals, then the few individuals that are remaining that are still susceptible to the infection are protected by the herd that's surrounding them. The protected herd is interrupting the cycle of passing the contagious infection to others. So you can see that while there were 3 susceptible people, on the left, when the infectious individuals entered the population, only 1 of the susceptible people got infected. The rest were protected. This illustrates the concept of herd immunity. With COVID-19 presently, the infection rate is becoming quite low, but there is still infectious individuals. However, the combination of increased vaccinations and previously infected individuals is beginning to produce the benefits of herd immunity. At present, herd immunity is not ideal, but hopefully more people will agree to be vaccinated and the herd immunity will be in place. Vaccination is going to be key to achieving herd immunity because the mortality rate is much higher than even with influenza virus. And the level of spread is faster. Very many more people would have to die of the infection in order to achieve the herd immunity without widespread vaccination. Obviously, to imply that we need to let the pandemic run its course and achieve herd immunity the natural way through infection, that's not really an ethical option if you really know what you're talking about and understand that the vaccine safety is not a real concern compared to the viral infection itself. So here's my brief PSA, and it's especially addressed to those of you in or going into the healthcare setting. You have chosen a career to care for the sick, immunocompromised, and other vulnerable populations. You need to do your part to ensure their safety. You have an ethical obligation to ensure that you are not the source of an infection that may devastate or take their life. When a vaccine is available to prevent you from an infection, you might also be protecting your patient population when you take it. You need to weigh a vaccine refusal seriously. Why exactly are you refusing the vaccine? You don't want a headache the next day? Well, your patients don't want to die because you coughed a virus you didn't really know you had on them either. Do your homework and don't be easily swayed by propaganda. You have the ability to really evaluate the science, so do it. Make a valid, thoughtful choice based on the reality of the situation. not emotional ones, as we tend to do when we don't stop and analyze the whole situation. Okay, done with the PSA message. Back to the chapter material. Let's move on and talk a bit about passive immunotherapy. We've already talked a bit about it. Recall that passive immunity is when an individual is given preformed antibodies. This is also called passive immunotherapy. The immune part is the antibodies of the immune system. And of course, it's therapy or treatment. The reason the antibodies are given is the individual needs immediate protection. Perhaps they've been exposed to a toxin, a venom, or are ill with an infection, and you're giving them the antibodies from a protected, immunized, or recovered individual in order to allow them to have protection as well, even if it's temporary. So, what are the limitations of this type of therapy? Well, you're not going to be able to purify out only protective antibodies if you're using what's called antiserum. Antiserum is the antibody portion of the blood. You purify out that protein portion of the blood, but there's going to be antibodies against lots of different antigens in that fraction. If you're purifying antiserum from individuals or even animals, sometimes that antiserum is going to trigger allergic responses, especially when they're coming from an animal source. This reaction is called serum sickness. It happens because the animal antibodies are foreign proteins and your immune system can detect that. For example, if the antiserum was made in a horse or a goat, and then you give those antibodies to a person, the first time you give it, it might provide good protection. However, your immune system is going to respond to those foreign proteins made in a goat. And so the second time you get that particular treatment, you can have an allergic reaction. The reaction termed serum sickness. Another concern with antiserum is that because you're working with blood products, you might have viral pathogens that are contaminating that product. This has happened in the past. Early on in the HIV epidemic, they didn't really know anything about HIV, and thus they weren't testing for it. HIV was unknowingly being transmitted when they were purifying blood components to treat hemophiliacs and others. These individuals were acquiring HIV infection from the medical treatment. Viral pathogens are of special concern and can contaminate the antiserum. Unless they're specifically screened for to reject contaminated blood, the viruses can be purified along with the blood components and be transmitted to the recipient of that treatment. Additionally, we know that antibodies degrade relatively quickly. They are proteins, and proteins are not long-lived molecules. They degrade, usually within a few weeks. Therefore, we know that passive immunotherapy is a very short-term fix for an immediate problem. Some of the concerns with passive immunotherapy are addressed by the use of hybridomas. I don't know if you recall, but when then President Trump contracted COVID-19, he was given an experimental therapy. While they didn't tell us too much about that therapy, we did know it was a monoclonal antibody-based therapy. Let's discuss how those work. First, we need to discuss the basic process of making a monoclonal, not one for therapeutic purposes. Monoclonal antibodies, shown right down here, is abbreviated MAB. Mono, meaning one, and AB, the abbreviation for antibodies. When you see a MAB designation on a drug therapy, it's a monoclonal antibody, but we'll come back to that in a moment. So we're talking about this scenario right here, mono meaning one clone of a B cell, right? One B cell making one type of antibody. We want it to make large quantities. How do we do that? First, you basically vaccinate a mouse against the antigen that you want antibodies to. So you inject the mouse, and the mouse's immune system responds and makes antibody. Their B cells are producing antibody. Then you're going to isolate those B cells from a mouse. Usually it requires taking the spleen from the mouse and isolating a large number of B cells. When you do that, you're going to need to make those B cells immortal, meaning they're not going to die after a couple of days in the lab. B cells do not typically divide more than a couple of times, and then they die in a lab setting. How do you make a B cell immortal? Is to fuse them with a cancer cell. So this type of cell that they're showing in this flask is a cancer cell called a myeloma cell. They live forever. Cancer cells, they just keep dividing as long as they're supplied with nutrients. They don't listen to normal cues to stop dividing. They're immortal in lab. There's a mechanism to fuse these cells together so you have a B cell and a myeloma cell that's been fused. Some of those cells die, but some of them actually make it through that process, and then they're a hybridoma. Now it's an immortal B cell that can make antibodies forever in the lab. It will live as long as you give it the right nutrients and room to grow. The hybridoma, it'll keep living, keep dividing, and keep producing antibodies for you. But you're going to have a whole bunch of different types of hybridomas in the mix at first. You're interested in finding the one cell type that responds to the antigen that you're interested in. To find that cell, you're going to have to dilute out those mixed hybridoma cells so that you essentially have a single cell in each well of this plate. They will grow and divide and produce antibodies. Then you test the solution in the wells because that's where the antibodies are going to be. You'll test the solution to figure out which of these wells has the B cell in it, making the antibody type that you're interested in. Once you've located it, as they've shown right here, that well had the B cell in it, making the antibodies you're interested in. Once you've located it, you can just grow the cells up in huge numbers and purify the antibodies that they're making for you. The antibody is going to be against a very specific antigen, and this is your monoclonal antibody. This is a classic type of monoclonal antibody. These types of antibodies have lots and lots of uses in lab. You can purchase specific types of monoclonals to perform tests in the lab. But as far as treatments go, if you wanted to give this monoclonal antibody to a person to treat a disease state, you're going to have to do something a bit different. Remember, we just talked about the fact that our own immune system, a human immune system, is going to recognize a mouse antibody as foreign. Mouse antibodies and human antibodies are not exactly the same. So, a mouse antibody is different than a human antibody. Enough antigenic differences exist between the two types of antibodies such that our immune system will recognize the mouse antibodies and make a response. The next time you try to give that monoclonal antibody, it's not going to work because our immune system is going to clear it out. To use monoclonals as a therapy, we need to do something different. We're going to genetically engineer the monoclonal to be what's called a chimeric antibody. Do you know the mythical creature called a chimera? It was a hybrid creature made of different animal parts like a lion body and a goat head. Well, we're going to discuss how to make a chimeric antibody. So what they do is take a gene. I'm going to draw an antibody figure. So we have the light chains and the heavy chains. They're kind of held together with some covalent bonds. Right there, but this portion right here, the tail end of the antibody, that part is referred to as the FC portion. We need to fix it to make it work as a treatment in humans. What they've done with genetic engineering is they've changed the gene of the heavy chains so that they can take the B cell of interest and manipulate the genes. The researchers can remove the segment of the genes that codes for the part of the heavy chain, the FC part, and replace it with that same section from humans. Alternatively, they'll now have what's called humanized mice. These mice are already genetic engineered so that the mouse has the human part of the FC antibody gene inserted in place of the former mouse FC heavy gene. When researchers go through this whole process of injecting the mouse and isolating the B cells will already be making antibodies that are human mouse chimeras. Antibodies made in one of these two ways, part mouse, partly made by the mouse immune system, and partly human genes. When you isolate the antibodies and use them as therapeutic agents, the human immune system will no longer recognize anything foreign and will not respond to it. This is monoclonal antibody production. It's really fascinating, and a lot of new drug treatments are coming out of this technology. This class of drugs that includes monoclonals is called biologics. If you see that term, they're talking about proteins used as treatments, and chimeric monoclonal antibodies are often used. Initially, you need mice to make B cells to respond to the antigen of interest. However, after you isolate the hybridoma, making the correct antibody, you only need the instruments and processes of the lab to produce the mass quantities needed for treatments. These new biologic treatments, obviously, they're proteins. Many of the protein drugs, these chimeric monoclonals. To administer a protein drug is a little different than administering other types of drugs, and special handling procedures will need to be employed to keep the drugs active. Developing and manufacturing these biologics is a very expensive process, and they currently charge quite a lot for these treatments, thousands of dollars per dose. If you want to know if a drug is a monoclonal biologic, look at the actual chemical name of the drug, not the brand name, like Humira. Look at the name of the drug and see if it ends in MAB. Humira is one, but the name of the drug is adalimumab. See the mab at the end? The monoclonal biologics all end that way if they are a biologic, a monoclonal chimera. Take a look around, especially those of you in healthcare setting. You will start to notice these drugs in use. I think you'll be happy that you understand what this is all about. They really are the new wave of treatments coming into the healthcare system. I think we better end this discussion here, and we can pick up the next lecture comparing passive immunity to active immunity.